Is 3:33. Welcome everyone to APS seminar. As usual, at the end of this seminar is when we'll take questions. So you won't have opportunity to take questions during the seminar. So you can type your questions into the Q&A box at any time throughout the presentation, and then I'll kind of handle them in the order that they're received at the end. Or alternatively, if you'd like to ask your question live, you can raise your hand and Cody will open up your mic and then you can ask your question directly. So our speaker today is Dr. Ratan Chopra. Ratan has been on campus for almost 4 years, but his background is that he got his PhD at Texas Tech University in Lubbock in 2014. His advisor there was Dr. Mark Burrow, and he worked on peanut. And then he did a postdoc for USDA ARS, also in Lubbock, with Dr. Gloria Burrow for 2 years working on sorghum. And then in 2017, Ratan came to the University of Minnesota and was a postdoc for David Marks until December 21st of this year, when he joined our project in the Department of Agronomy and Plant Genetics. And we hired Ratan partially to take some of the responsibilities of Dr. Catherine Frells, who just accepted a position as wheat breeder at the University of Nebraska. And today happens to be Catherine's last day. I'm sure she's in the audience today. So congratulations again, Catherine. And very happy to have Ratan join our project, though. He's been very productive in research, grant writing, publications, patents, mentoring students, working with other researchers. We know him well. So he's going to give us a little bit of his background, talk a little bit about peanut, sorghum, but the majority of this is going to be on his pennycress work since he's been at the University of Minnesota. So with that, Ratan, I'll let you share your screen and take it away.
Thank you, Jim. I'm just gonna verify if y'all can see my screen. Can you see my screen?
It was there.
Okay, let me try again. How about now?
There you go. That's full screen.
Okay. Thank you, Jim, for the introduction. I'm Ratan Chopra. Today I'm going to talk about trait discovery towards increasing the land productivity, and I'm going to give some examples from sorghum and pennycress, for which I've worked on for last several years now. With that, so before I get into my research over the last few years, I want to give my introduction a little bit in detail. I started my research career as an undergraduate back in India, working on velvet bean, where I was involved in looking at phenotypic and genotypic diversity among the germplasm collection of this velvet bean in India. That kind of helped me develop an interest towards molecular breeding or in general molecular genetics work. So I joined program at Texas Tech to work on peanut genetics and peanut genomics. After that, I continued for my postdoc at USDA ARS. And during that time, I was still looking at the genetic diversity, but I picked up interest in abiotic stress tolerance, specifically early season chilling tolerance in sorghum. And while I was working on that, I also was involved in looking at traits in sorghum using mutation approach. Um, After a couple of years, I had this wonderful opportunity to come here at University of Minnesota to work on winter oil seeds. And I have continued to work on all these three aspects, whether it's developing genetic or genomic resources, looking at how changes in temperature or weather patterns can affect the pennycress development, or the traits that would help us domesticate this new crop. With that, um, my overview of my presentation would be I will highlight briefly about my PhD project and then get into the specific for a couple of projects in sorghum that I worked on. One was towards chilling tolerance in sorghum, and the second one dissecting the yield components. And then how that knowledge that I gained in sorghum, I was easily able to translate that into pennycress and utilize that knowledge towards developing the resources and domesticating this new crop. So I started my PhD with Dr. Mark Burrow at Texas Tech University. Um, and during that, I was assigned a project to look at both the phenotypic and genotypic diversity that existed in the wild and the tetraploid collection. So in the wild collections, peanut generally looks like pretty small. The pod size is very small, the seed size is very small. And then there was a wild tetraploid as well. But in modern cultivars or the football peanut that we enjoy every weekend are usually bigger in size compared to these diploid peanuts. So there was this huge phenotypic diversity between the wild peanuts and the tetraploid peanuts. So I started looking at nucleotide diversity among these collections using a transcriptome-based SNPs. And what I found was I could cluster A genome accession together, K genome accession together, and B genome accession together. But all the tetraploids kind of overlapped each other. Whereas the synthetic tetraploid that was derived from the cross between these two was in between these two groups. On further inspection of these tetraploids, I was able to cluster out the fastigiata type and the hypogia type. Basically, was able to separate out the erect type or the runner types. And this wild tetraploid, which was merged here, was clustered all by itself. So this suggested that the uh, SNP that I used to look at the diversity in this collection was useful. So I decided to utilize that SNP and develop a linkage map for a diploid peanut population and look for QTLs for several plant architectural traits. And the idea here was that the population that was used for this mapping population was showing differences in some disease resistance. And I wanted to see if I could utilize these markers and develop a linkage map. And in the future, somebody could look at these disease resistant traits in this population. As I finished my PhD, I was I joined right across the street at the USDARS location with Dr. Gloria Burrow to work on sorghum. And specifically, I worked on early season chilling tolerance and then some of the yield related traits. So before I get into the results, I want to talk about why chilling tolerance or how is that an important trait in sorghum. So sorghum is generally produced in the high plains area, and farmers would like to plant it early in the spring or like in the month of April to kind of utilize that ground moisture that is available from the winter time, and also kind of overcome those drought period or dry period that occurs in the month of June to mid-June, which affects the grain filling in sorghum as well. Um, and the other important issue that we can overcome with planting early with recently over last 7 to 8 years, there has been increase in production sugarcane aphid pressure, and by planting early, we can kind of overcome that issue as well. So when I say early chilling, early season chilling tolerance, the difference is here. Like you see on the right side, that's a susceptible line, and the tolerant line is more vigorous. So basically, we wanted to find lines that would be vigorous in the early season, even with those fluctuating temperature. And to screen this trait in the field condition, it's very challenging because the weather patterns are not predicted from year to year. And when you want to select for a line, you have to do multi-year, multi-location testing, which is a lot of uh, dollars invested in the field trial. So my role during that tenure at USDARS, I was looking at, can I develop a technique or a tool to screen for germplasm that would help us identify these early season chilling tolerant line. So to do that, I used an integrated approach. So I started by first looking at the association panel that was put together back in 2008, for which GBS data was already available. And I took that association panel population, looked at stress responses at the seedling stage under cold stress. And we looked at several traits: shoot weight, shoot length, root length. And importantly, we looked at the anthocyanin content in the leaves under cold stress. And there was good variation for all of these traits. And I decided to run association mapping because we already had the GBS data available for this panel. So upon running association analysis, what I found, there were a good number of markers associated with this trait for the anthocyanin levels in the leaves. So basically, this plot explains, this is a Manhattan plot showing the chromosomes on the x-axis and the significance on the y-axis. And the green dot represent the significant markers that I identified. So when I found those markers, I started exploring for nearby genes. As the sorghum genome was already sequenced, well annotated, I started exploring genes in nearby these markers. What I found on a close evaluation, one of the markers were very close to this anthocyanin regulator, BHLS transcription factor. And this was very encouraging because we tried to map for anthocyanin trait in response to cold in sorghum, and I'm finding a gene that is actually an anthocyanin regulator. So it was very exciting to me. So I decided to further explore this co-expression network that existed for sorghum. So when I looked at this expression network, I focused very heavily on this part of the network because I saw there was a consistent, most of these genes were involved in sugar metabolism, sugar biosynthesis, and sugar breakdown. And because of that, I decided to do qPCR on a lot of these genes between the tolerant and the susceptible line. And what I noticed that always the tolerant line had a higher expression of a gene compared to the susceptible line. When I saw this, I decided, well, if we are seeing differences in the sugar level, so why don't I perform HPLC, just a simple sugar analysis in the seedlings, and see if I see differences between the susceptible line and the tolerant line. So what I did, I grew these seedlings out. After 7 days under cold, I took the susceptible line, the tolerant line, and dissected those seedlings and ran HPLC. And what I noticed that the susceptible line, for example, TX7000, and the tolerant line Mucin Z, uh, showed differences in the amount of fructose. I went back to the field, looked at the seedling vigor and the root length and the shoot length, and I saw differences between the tolerant line and susceptible line as well. So this kind of made me think, well, not only we have the genetic marker, but we also have a metabolite marker that could help us select the germplasm for early season chilling tolerance in sorghum. So then I decided, well, I'm seeing this as a potential metabolite marker. I want to plant lines that are known to show variation for cold susceptibility, and also the lines that are shown to be tolerant lines. So I planted about 30 lines, and I did this bioassay in the lab after 7 days for those 30 lines. And I grew these lines out in the field. And in the field, I took notes like germination and vigor rating, whereas in the lab conditions, I looked at all the simple sugars and some of the complex sugars like durin and another polysaccharide. And what I noticed that there was a positive correlation between the field traits and this metabolite that I identified. You will notice that this is negatively correlated here, but actually it's a positively correlated. The way I scored the vigor that time was on a 1 to 5 scale. So because of that, it's a negative here. But generally, this gave me a confidence that not only we had the DNA marker of interest, but we also had a metabolite marker that we could use for selection. So I was again curious. Once I identified that potential marker, I wanted to see, can this be applied for selection of a line from an inbred population. So I had access to this inbred population, about 300 lines in the inbred population, and I grew those lines out in the field and performed HPLC analysis on all those 300 lines. And what I found, there were a handful of lines here that had good germination, good vigor, and good fructose level. And more importantly, I wanted to focus on shorter plants because of the grain sorghum. Usually, you want a dwarf plant. So using those criterias, I was able to successfully identify a couple of lines. I worked with Dr. Amendak at USDARS location, who's a physiologist, and he evaluated those lines. And he was able to confirm that these two lines were not only early season cold tolerant, but they were also having pre and post flowering drought tolerance, and they also had a larger seed. So that was my cold tolerance work that I did during my tenure. But while I was working on the cold tolerance, as a part of the team, I was also involved in the mutation breeding program that was ongoing. And the mutation breeding program already had population developed from 2008. But when I joined, what we were doing, we were trying to build the mutation library for this mutant population. So as a part of that project, we sequenced over 250 lines, and we looked at several traits, and I was specifically working towards like bloomless, which is a reduction of epicuticular wax, brown midrib for forage quality, and then yield components such as multi-seeded sorghum and increased seed size. And today I'm just going to talk more about the multi-seeded sorghum and the increased seed size. So when I joined the program, they had already had the population, like I said, but they had identified numerous mutants. And one of the mutants was multi-seeded sorghum. And why that is in a wild type sorghum, what happens is you have one sessile and two infertile pedislates. Sessile is fertile, but the pedislates are not fertile. And in that mutant population, they found a line that could produce a fertile pedislate as well. So as a result, what made everybody think that you could have basically three seeds instead of one seed, so you could increase your yield in sorghum. So the work was done towards identifying genes that were responsible for this trait, like TCP, LOX2, FAT7 were already characterized when I was there. And what we noticed that most of these genes were involved in jasmonic acid pathway, which kind of makes a lot of sense because JA pathway does affect the flowering or pollen viability. But what was interesting to me is when you increase the seed number, we were noticing the seed size was relatively smaller. As a result, even though you have number of seeds increased, but your net gain was not necessarily huge. Your net gain was minimal. So my interest was when I looked at this particular trait, what I wanted to do is I wanted to explore if we could identify bigger seed size in sorghum. So to do that, I started surveying the literature, went back to QTL studies that have been done in sorghum, rice, and maize. And I was specifically looking for those QTL regions and seeing if there are some genes which carry mutations in the mutant library that was existing at the location. And I found several genes, and specifically the two I'm going to talk about are the genes that were identified, or the QTLs that were identified in Dr. Patterson lab in Georgia. So I found mutations in these two genes. I backcrossed these lines. And what I was looking for is if these mutations would have, what kind of effect they would have on the total panicle weight or the 1,000 kernel weight. So the first gene showed that it had a negative effect. So, and when I was doing this, I didn't realize that it was a sweet 4C gene. Sweet 4C gene is actually known to reduce the seed weight when you have a mutation. And this is a very important domestication gene that has shown that in maize and rice that contributes towards increased seed size. Whereas the other gene, I noticed that there was an overall positive effect on the panicle grain weight, total panicle grain weight. All of these, so what I suggest is basically you could retain some of the favorable alleles in the background that you use for mutagenesis, but you can also carry over some of the alleles from the mutant population to increase the seed size. So with all these mutations identified, or the causative nature identified, we could cross it back to the multi-seeded sorghum and increase that overall seed size, which is an issue. So having talked about these two projects, I'm going to summarize by saying that, yeah, when we want to understand a complex trait like chilling stress tolerance, it is important to integrate multiple genomic approaches to identify these, either the metabolite marker or the DNA marker, and you need to perform mark crossing, and you can validate, establish causality between the gene and the phenotype. What is also something that I learned during that 2 years is it's not only about looking at a germplasm of from like just the natural accession. Sometimes it's important to combine both the natural accessions and the mutant population to accelerate the development of new elite breeding lines, or identifying the alleles that would be crucial for any breeding program, or to overcome any challenges that could occur in sorghum or any other crop. So when I was wrapping up this work, I was actually attending a meeting at plant and animal genome meeting, and I ran into Kevin Don, and I came across this opportunity on pennycress, and it really sounded very interesting to me. So because I already had this experience in mutation breeding and how that was being used towards developing traits in sorghum, so this was a very natural fit for me to make a move up here. And when I joined the program with Dr. Marks, I was assigned the role of looking at genomic data, identifying traits that would be responsible for domesticating this new crop. So before I get into the domestication of this new crop, I want to talk a little bit about what pennycress is. Pennycress is a winter oil seed that belongs to Brassicaceae family. It's a diploid, it's a self-pollinating species. The genome size is about 500 megabases, and then it is a short life cycle and can produce up to 1,000 kilogram per hectare. What's more important to this is pennycress is very closely related to Arabidopsis, and to an extent that even the gene space between Arabidopsis and pennycress is highly conserved. And pennycress is just one of many crops that are being worked under Forever Green initiative under the guidance of Dr. Wise. And what I'm going to talk about, all the traits or the improvement or the germplasm that is being developed around this trait discovery program, is on the line collected by Dr. Wise, MN106. And rest of the presentation is going to be based on that material. So what is domestication? I mean, domestication is basically genetic modification of a species to create a form to meet human needs. Depending on the crop that you're working on, you can make changes for your needs, and you can call it as a domestication. But there is like a classical traits that have been classified as a domestication syndrome, whether it's apical dominance, loss of natural seed dispersal, loss of seed dormancy, changing the photoperiod sensitivity, or synchronized flowering. And this is not just very new. It has been happening for all of these modern crops that we've seen, whether it's maize, where it's completely changed the species. In rice, the shattering issue has been resolved. Wheat, tomato fruit size has been increased. So there is a lot of these modern crops have gone through this alteration, resulting in like domesticating these new crops. And what has been nice over the years is sequencing technologies have improved, and people can identify DNA markers and use their breeding program to integrate into their elite lines. Having said that, what I told you earlier in a couple of slides, that pennycress is like Arabidopsis, and we can identify those traits. So here is an example like how Arabidopsis can guide our case. So Arabidopsis is well-studied model system for last 3 decades, and a lot of this pathway has already been characterized. You have mutations on the left side of this pathway here that can affect the fruit size. For example, fruit full or AP1, AP2, or R6 and R8 can affect the seed size or the fruit size, whereas mutations in these genes like IND, Shatterproof, ALC can affect the dehiscence zone, basically as a result of cell wall changes. Using this information, we knowing that we have pennycress as Arabidopsis, what we are doing is something different. Historically, what has happened is humans have done selections based on their needs and developed the cultivars or the varieties, and then geneticists have identified markers for it, and developmental biologists have tried to understand the genes and characterize them at the cellular level. But what we are taking is a slightly different approach. We are still trying to domesticate a crop, but we're trying to accelerate that by using the knowledge of Arabidopsis, using Arabidopsis as a tool instead, and then create a variation in the genes that can help us domesticate this new crop, and then work towards establishing that causality and developing cultivars. So what I'm going to talk today is going to be mostly based on this approach. To do that, to induce variation in pennycress, Dr. Marks back in 2014-2015 decided to perform chemical-based mutagenesis in pennycress because of it is a diploid self-fertilizing, and knowing that the gene space was very similar to Arabidopsis, you could get Arabidopsis-like phenotype. He decided to perform mutagenesis using EMS, and thousands of plots were grown out in the field, and hundreds of plots were grown, thousands of plants were harvested. And using this resource, when what was found was basically you could see not only the gene space between Arabidopsis and pennycress was similar, but you could see phenotypes basically being copied from Arabidopsis to pennycress. For example, here, like you have elongated hypocotyl changes in the phyllotaxy, like the leafy phenotype, or even the changes in the seed color. When we found this variation, we did whole genome sequencing of all of these lines. In a matter of time, what we were able to do is clone this pathway, which is well studied in Arabidopsis in pennycress. We identify genes that are responsible for all these seed colors in pennycress as well. So using that information, knowing that pennycress is similar to Arabidopsis, I'm going to talk a little bit about traits now. So one of the traits is reduction of natural seed dispersal. Uh, so any modern cultivar or any major crops today have gone through this process of reducing the pod breakage or reduction in seed dispersal. And that is what differentiates a wild species from a cultivated crop. As you can see here on the right side, that all of these modern crops, whether it's soybean, tomato, rice, sorghum, all of these have gone through these changes in the cell layers, affecting either the breakage of the pod or dehiscence zone in these grains. And similar problem exists in pennycress. In pennycress, we see a natural seed dispersal because it's a weed. And because of that, you can see after harvest maturity, every week you could be losing certain amount of pods. And after about 4 weeks, you could pretty much lose 90% of the pods to the ground. And because of this issue, you really have to resolve this issue before you can make pennycress as a crop, because you can on average have about 60% or more yield reduction. So it's not going to be feasible for farmers to lose that much amount of seeds, but also you create a seed bank that's going to compete with the main crops in the following season. So we went back to the mutant population to look at the variation for this reduced shatter phenotype. And what we were able to do is identify 6 lines that had this reduced shatter phenotype. This is a wild type that shows that the pods are shattered after harvest maturity, but we have mutants that do not shatter the pods. And so I decided to sequence all these 6 lines. And what I found was there were mutation in gene called indecent1, SPT, PID. And all of these genes are very well studied in Arabidopsis. And what I was what was interesting to us is we found that 3 lines carried a mutation in this gene called indecent1. So having these three independent alleles confirmed that the mutation indecent 1 is causative gene in pennycress. And in pennycress, like in other species, there was a change in this cell wall development. We saw the same change in pennycress as well. So using that information, and we were able to identify these lines and characterize them. But what is the impact of having this trait in pennycress? So I decided to plant these in the field and then let them uh, after maturity, I just let them stand in the field for about 4 weeks after maturity. And I was taking notes every week to see how many pods are retained after every week between the wild type and the mutant line. And what I noticed in the wild type, we saw that there was a huge reduction or loss of pods, whereas the mutant had retained most of the pods. Although I'm not sure if the picture are clear, but the wild type has lost most of its pod, but whereas these mutants have retained most of their pods. So that's an impact of having a reduced shatter line in any in pennycress. Like shattering, the other issue for any wild species or any weed to domesticate is a seed dormancy and having a uniform germination. All these modern crops are viable because of this uniform establishment and uniform growth. And if this to make pennycress as a viable crop, we want to have that uniform germination, and we should reduce any primary or the secondary seed dormancy that exists in pennycress as a weedy species. But having said that, this is a very complex trait. When you want to solve the seed dormancy or germination issue in pennycress, uh, there's a lot of factors that can affect this process, whether it is the seed coat or the optimization of the seed composition. For example, like just changing the fatty acid profile of pennycress seeds might affect the germination, or the environmental conditions that would affect the seed maturation. Like during maturation in like month of May or June, you could have really warm spell or you could have a cold spell. Does that affect the seed quality? You could have issues with the germination with water stress in the fall. Some years we have a nice wet fall. Some years we have very dry. Like last fall was very much dry for a while. And then more importantly, a lot of the modern cultivars are successful today is because you can bury them a little deeper to capture that moisture. And pennycress, we want to do the same thing. So we want to plant them. So it's a complex trait. And it has been shown even in Arabidopsis that there are a lot of genes affecting a lot of different parts of the puzzle here for seed dormancy or germination. So we've solved some of this, and I'll give a couple of slides on that. So the first thing we looked at was if we modify these seed codes in pennycress, do we improve germination or improve seed dormancy? And what we saw, indeed, having this yellow seeded line, we could increase the germination compared to wild type. Like this particular line, this yellow seeded line was planted 3 different seasons. And every time what we noticed that the transparent tester line outperformed the wild type in terms of germination without any treatment. Fall 2019 was a very dry year and we did not get enough moisture in the fall. So that's why we see such lower numbers. But generally, we see the trend that TT2 line always germinated better compared to the corresponding wild type. But if we did treatment like priming with water or GA, we could improve that germination in the wild type, but definitely we could have better germination even in the mutant as well. So this was very encouraging. So we could have uniform and faster germination using this yellow seeded line. But last year, when we had hundreds of plots out on St. Paul campus, uh, we decided to leave those plots longer after the harvest maturity. And what we saw that a lot of the pods shattered, and I went back and collected notes in those plots. And what I was looking for, are there any lines when they shatter, do they right away germinate and establish, and how vigorous they are? And I made a scale of 1 to 5. And what I was looking for is if the lines germinated completely and are robust, then I gave them a rating of 5. So what I noticed that the 106, which is a check line, had a score of on average 3, but these all these other lines had much better score in terms of vigor and establishment. Going back and looking at the pedigree, all of these lines had the yellow seeded trait in them, which suggested that having yellow seeded line would not only help us improve the germination, but also help us get rid of the fresh seed dormancy that may exist in pennycress. So that is just one part of the puzzle to solve in terms of germination or seed dormancy. But what we want to do is try to get rid of all the weedy characteristics of pennycress. So the other thing we wanted to remove or resolve is germination under low water potential. To do that, we developed an assay in the lab in a Ziploc bag. We created this water potential, try to create this reduced water potential using PEG 8000 by creating several different concentration and screen some of the mutants that we knew could be a potential candidate. And we found this BZIP67 line that germinated even at 25% PEG, where the wild type did not germinate and the other lines did not ever crack open, even after like 15 days. So this was encouraging to us that we have a line that could germinate even at a very low water potential. Then same lines that we had as a candidate for good germination under low water potential or in the field condition, we decided to plant them at 3 different depths in the field conditions last year. So to do that, we used a push planter and planted at 3 different depths, 0.25 inch, zero point 0.5 inch, and 1 inch. And after about 14 days, what I noticed was there was this line derived from Somnus gene that germinated way better than any other lines at that higher depth. So we have identified lines that germinate at a greater depth. We've identified lines that germinate at low water potential. And we've identified lines that germinate uniformly or better than the wild type. So now we are in the process of combining all these three and hoping that we can solve some of these reduced seed dormancy issues or germination issues in pennycress. So those are very important agronomic traits which will make pennycress as an attractive crop, both for an agronomist and farmers. Once we have those resolved, we want to have a market for pennycress as well. As a wild species, as a pennycress, contains a lot of detrimental compounds. For example, oil contains high levels of uric acid, which is not good for human consumption. Uric acid is good for biofuel industry, but if you want to create market opportunities beyond biofuel for pennycress, then you need to get rid of that. And similarly, if you want to use pennycress seed meal for food purposes or feed purposes, there is a compound called sinigrin, which is a glucosinolate, and a lot of all the brassicas do produce glucosinolates. But pennycress produces glucosinolates in the form of sinigrin, which is detrimental for feeding the cows or dairy industry. So there are a lot of these compounds we want to get rid of to be able to market pennycress meal or pennycress oil. So we went back to the same population that I showed where we found the variation for shattering phenotype, variation for dormancy. We looked for variation for fatty acid and glucosinolate content using the same population. So we used NIR and looked for variation, and we found huge variation for all of these fatty acids like oleic acid, uric acid, linoleic acid, and also we found for a variation for glucosinolate content. This was encouraging. And I'm going to give a couple of problems that we've already solved in pennycress using this mutant population. The first one is modifying fatty acids in pennycress seed oil. So pennycress seed oil contains a lot of uric acid, like I mentioned earlier. Um, so we were surveying that population. We found a couple of lines that had complete loss of uric acid, or it did not produce any uric acid. Upon sequencing these two lines, we found that there was a stop codon in a gene called fatty acid elongation 1. As a result, you block the production of uric acid, so everything is backed up here. So we saw an increase in oleic acid, linoleic, and linolenic acid, which is interesting to see, but having this high levels of linoleic and linolenic acid can be detrimental if you want to use it for food application, because this can affect the stability or the storage of the oil for a long time. So we started exploring for variation in linoleic and linolenic content, and we started finding some lines that had reduction in linoleic acid content. We found several lines. Two lines had a mutation in gene called reduced oleate desaturase 1, which had about 30 to 40% reduction in linoleic acid compared to the wild type. We did find some lines that had complete reduction, but that had a mutation in a gene called FAD2. And whenever we have a mutation in gene FAD2, a complete loss of function makes the plant susceptible to cold, and they are not anymore winter hardy. So we wanted to find lines that are winter hardy and can have reduction in this polyunsaturated fatty acid. So we found these two lines, FA1, ROD1 mutants. We decided to combine those, and we were able to create this profile for pennycress that is similar to high oleic canola. So now we have a line that is high oleic, as similar to canola, and doesn't have uric acid. Similarly, glucosinolates, again, very well studied pathway in Arabidopsis. But what's different in pennycress is we are just looking at aliphatic derived glucosinolates. And this particular pathway has been studied in Arabidopsis, but a lot of the genes uh, are not characterized in pennycress. And we were able to characterize some of these now. And the most important thing I want to bring your attention to is a line that we found where there was a 90% reduction in total glucosinolates. And there were, upon whole genome sequencing this line, I found a mutation in a gene called AOP. uh, this AOP gene reduced the glucosinolates in the seed meal. But what was puzzling to me and David was this gene was downstream of the pathway. And having a mutation in downstream of a pathway can affect this glucosinolate to this level. It was, we were not expecting that. So we did co-segregation analysis, found that this mutation was tightly linked to the phenotype. Then we decided to do HPLC on this. And what we found that wild type produces this high levels of synegrin, but this AOP mutant does not produce synegrin. Instead, there is a backup product that is a precursor for producing this synegrin levels. So we've been able to modify oil content, we've been able to modify meal quality, and we've identified several agronomic traits. And what we've been able to do is not only identify these traits, and we've been able to combine these traits into a single line. Here is an example showing a high oleic reduced shatter line that we've been able to develop over the last few years using the traits identified. And then what we've been able to do is basically go from a stinkweed to an oil seed by removing this glucosinolate or reducing the uric acid. And right now, we have these quadruple mutants growing out in the field. Here is the wild type, and here is this picture is from this past fall of 2020. We will know how they perform in terms of yield, but we are making good progress in terms of domestication. We've stacked all of these traits I talked about, and hopefully we will get the yields that we need. But having said that, what we have done so far is basically identify these foundational traits that would help the breeding program in the future. We've identified these traits. Now the breeders can help us kind of get those yield gains through the traditional breeding methods. Having said that, I just want to give you a perspective of what has been done between rapeseed and canola. So pennycress, historically, like rapeseed was converted to this double zero by rapeseed also had like issues like uric acid, glucosinolate, and it took them several decades to go from rapeseed to canola. But what we've been able to do is go from performing the same changes in pennycress in a matter of a decade. And that's simply been possible because we've been open to ideas. And what we've done is we've combined traditional breeding methods such as mutagenesis with some modern tools like sequencing. And combining those, we've actually accelerated that process. And we are at a place now that we have this high oleic reduced shatter lines that is being tested at several locations this year. not only in Minnesota, but in Illinois, in Ohio, in Wisconsin. So we are testing this high oleic reduced shatter line for its performance as a part of the larger pennycress efforts. When So that is the status of domestication and what we've done over the last several years. But during that process, like I mentioned earlier, that pennycress is like Arabidopsis. And although 3 decades of research has answered a lot of questions in Arabidopsis, but there is a lot of questions that remain to be resolved. Uh, and what pennycress provides an opportunity is not only you could do that translational research from using Arabidopsis to pennycress and answer the same question in a different brassica, but instead you could kind of answer the questions for the genes that have not been studied in Arabidopsis or the genes that have been studied in more robust field conditions. So that's where we think pennycress can be a model system which can supplement Arabidopsis and also help other brassica crop improvement efforts. So to do that, what I have done over last 3 years is focused on creating this mutation library for pennycress. And I've already sequenced over 500 lines, and I've identified several million mutations in these lines. And our target is that we want to find multiple alleles in each of the genes that are predicted in pennycress. Already like, and we are on track to sequence another 500 lines. So our goal is to sequence 1,000 lines so that we could have enough alleles in each of these genes. And already looking at these 500 lines, we've seen that we have about 5,000 genes with a complete loss of function. So that's very exciting that we can use this resource for basic biology as well. What is the advantage of using this? Like you can say, well, it's close to Arabidopsis. You already could do translational research using gene editing, but we see an advantage of using random mutagenesis uh, in pennycress for both crop improvement and developing it as a model system. Because using this EMS approach, we can create a lot of alleles. Some might be weak, partial loss of function, some might be complete loss of function. And having these different alleles can help you understand the biology more clearly. For example, like we had these two traits that I talked about. One is the reduced shatter and the other one is early flowering. So having a complete loss of function in this gene can result in complete loss of this layer. As a result, you cannot break open the pod. So even if you have a reduced shatter phenotype, you could run a combine through, and then you have all these pods that are intact. So you have to do a lot of post processing. So that's not efficient. In terms of biology, you could have a mutations in this gene called ELF6. A complete loss of function would result in loss of vernalization capacity. And we had a partial loss of function. It gave us the right phenotype, optimal phenotype that could be used for the breeding program or the crop improvement, but also help us understand the biology of it. So not only using that EMS is good for crop improvement or as a resource for the model system, what I see this resource as an opportunity to train next generation scientists. Uh, you have this resource, you have these genes and phenotypes that you can use for translational research and show students, like here is an example. There was a population that was segregating for plant height. We knew what the gene was. We could basically go back and look at those genotypes and phenotypes in that population, characterize that population, and give this as an example for students to think about how you can go from a gene to a phenotype, or the other way around. You could look at the phenotype and do some bulk segregant analysis and identify the genes. And I've been very fortunate that over last 3 years, I've been able to utilize this resource and train a lot of students. And I'm just showing a few of them here. Is I can use this resource, try to make them think that you could use this resource to look at a gene and how that mutation can affect the phenotype. So it's a very good resource for training students. And I think it's a big advantage as well. Having said all this, where we are with this resource is actually helping a lot of efforts for pennycress improvement. Already, we are collaborating with a lot of folks within the university and outside the university, where we've put together large teams like I Prefer team, which is a USDA NIFA cap grant, where we the group is utilizing this resource to look for variation in the oil content, dormancy related traits, whereas recently funded grant through the DOE, which is also called iPrep, where we are looking at utilizing this resource towards identifying resilience trait. And over these last 6 months, looking at the capacity of this resource and what we've been able to do, we have got some interest from Arabidopsis Resource Center and Europe's Nottingham Arabidopsis Stock Center to host this material for biologists to characterize these mutants. So with that, I would like to summarize by saying that we've identified and validated several useful phenotypes and traits in pennycress. Our trait introgression is on track, and we have several quadruple mutants right now growing in the field. We are sequencing over 1,000 lines for mutants. We are sequencing another 500 natural accessions. The picture you see on the background is basically these are the 1,000 mutant lines growing out in the field this year, and this resource will be available for public in the near future. And seats will be hosted at 3 locations, and JGI will be hosting the sequence data for this. So I want to end by saying one thing, that we've done a lot of progress in terms of identifying traits that are necessary, or foundational traits that are necessary for pennycress, and now we'll work with the breeders to kind of improve those yields. But what we are also preparing is preparing ahead of the game, that we want to identify and resolve these issues that may come up in the future. Like, already over growing pennycress, we've started noticing a lot of challenges that pennycress can be susceptible to these abiotic stresses. For example, like in the fall, if you don't have good moisture, you can have poor establishment. In the spring, you could have flooding and rotting of plants. If you have an early thaw or things like that, you could have heat waves and fluctuating temperature that could affect the pod development or poor pod fill. As you can see that, I mean, last 3 years have been like, every year has been unpredictable. Like this year, we haven't had the whole January, we did not have a negative degree day. So it's kind of like hard to predict. So we want to be prepared any challenges that may come up in the future. So as a result, we've put together this team led by Dr. Sedbrook from Illinois. What we are trying to work on is utilizing that genomic resources that I'm developing and identifying lines that would help us resolve these resilience traits in pennycress. In addition, we are working both of these grants, whether it's the DOE grant or IPRO, these are over $10 million dollar grants that have been funded to kind of resolve not only the existing problems of pennycress, but also looking ahead and try to solve those resilience traits that are important to make pennycress successful crop in the future. So JGI is supporting, mostly I'll be working towards utilizing the support from JGI to sequence all this material, provide the material for studying, and I will be specifically working on understanding pennycress germination under reduced water potential. And the picture I show here on the left is the idea of the team is, once we identify lines that perform well under all these conditions, we are going to grow them at 3 locations under this farm bot system and look at developmental changes in our candidate lines. So that's going to be the future that what I'll be focusing on, especially towards pennycress. So with that, I would like to acknowledge and thank a lot of collaborators and colleagues. I'm fortunate to have worked with so many awesome collaborators and colleagues over the years. And I will take any questions you may have. Thank you.
Right. Thank you, Ratan. Good. We'll let you catch your breath for a second. We do have a couple of questions for you. So I'm going to go to the chat first, where John Hawkins asked, The multi-seed mutation reducing size resembles the situation of barley, where plants with 6 rows of fertile flowers show smaller seeds than those with 2 rows. In your opinion, is the reduced seed size due to division of photosynthetes across more seeds, or due to physical constraint of seed size by neighboring seeds?
My observation is with the constraint, the dimensions of the panicle affects the growth of those pedicellate seeds, and I kind of was able to verify that. But moved to Minnesota by then, um, and I think that's the reason. The physical barrier is the reason for those smaller seeds. So to see if that was true, I crossed that line to a open panicle line, and what I saw was we could have those larger seeds even in the pedicellate seeds, and that's the picture. I Sorry, I can share that picture. Sorry, I'm just going to go back and show that first line. Yeah, so this is the line that I was able to identify by crossing a open panicle line of sorghum and this multi-seeded line. So this kind of gave me an idea that it was more of a physical barrier.
Okay, thanks. And another question also from John Hawkins. There is a theory of domestication that claims that germination from greater depths associated with greater seed size. In your experience, is this case in pennycress?
Uh, Definitely, it would be the case. We are in the process to test that if increasing the seed size would help us plant deeper. But what I can tell, that line that we identified that germinates at a greater depth has about 10% bigger seed.
Okay, and from the Q&A, Sunil Raju, great and really cool work. You mentioned that gene order is conserved between Arabidopsis and Ecress. I was wondering if you can comment on the level of conservation at the cis-regulatory sequences in promoters and enhancers.
Um, we haven't necessarily evaluated like the conservation between the promoter elements yet, but at the gene level, we've seen that about 70% of the genes are almost 80% or more similar at the protein level. At the protein level, about 70% of the genes. We haven't necessarily looked at how the promoters are conserved between these two species.
Okay. And Catherine Freckles is asking, how will you measure pennycress success 10 years from now? What traits will be key to that success?
Oh, I kind of alluded that somewhat in my talk, that what we've done is identified these foundational traits for pennycress now. Now we can go back to the breeding population and try to use these foundational traits, and hopefully we will have varieties and cultivars in a decade that plant farmers can plant, and there is a market for pennycress. That's the hope. Now we have the foundational traits, so I'm hoping that we see a good step forward to making pennycress as a successful crop.
So of traits that you mentioned, which do you think are the most key for domestication?
Uh, Definitely having a reduced seed dormancy, good germination, and reduced shatter would be the primary traits. Definitely having low heuristic would be good if you want to go towards the food side, food industry. But the agronomic traits is what is going to make farmers attractive to the crop.
Okay, thanks. Gary Muehlbauer, where is natural population collect from that you are sequencing? And is there an expectation or empirical data that there will be lots of genetic variation?
Uh, so until now, most of our collection was from Northern America. But over last 6 months, as we've got people interested from Europe and Russia, and so John Sedbrook out of Illinois State has led this effort of collecting germplasm from different parts of the world now. So we're hoping that we can capture a lot of variation. We have some information that there is a lot of variation in Eurasia. So we are reaching out to partners in Russia to see if we can get that collection. Hopefully, we can capture a lot of variation using that population. We just have representation from Armenia at the moment, so we cannot tell that how much more variation we can capture with this new collection. But what we are doing to make those selections for these 500 accession to be effective, as we are bringing in this germplasm, we are working with Oak Ridge Lab. And what they are doing is basically looking at those microclimate environments where these lines are collected from, and then going back and looking at Arabidopsis, seeing if there was variation coming from those regional areas for in Arabidopsis. And based on that, we are going to pick up those lines for sequencing in the future.
Okay. From Roger Becker, any speculation that evolutionary decreases or increases in sinigrin production would have altered pennycress plant performance as a weed, or in your case, will alter seed production and yield or
Uh,
other effects of lowering?
Definitely, that's a good point there. We are already seeing that issue when pennycress is definitely evolved to retain that glucosinolates in its natural form. And as and when we are changing these glucosinolates, we are noticing that there is issues with the pests in the field condition. Also, we are seeing some effect on the seed yield. Uh, I cannot necessarily say that it's completely due to glucosinolate at the moment because these are EMS lines. So we might be having some background mutation that's affecting yield, but we are looking at both the CRISPR alleles and the cleaner EMS alleles to see if these glucosinolates, how much they impact. They definitely impact. We've seen that reducing glucosinolates decreases the plant height, which decreases the yields overall. But we don't have that exact number, but we do see some reduction for sure.
So can you maintain glucosinolate levels in the plant material, but stop it from being loaded into the seed?
Uh, Definitely, we could do that. But what the genes that we've gone after as a collab with John, what we've noticed is when we try to knock those genes that are already characterized in Arabidopsis, we see that affects the plant or the flower development. So we have to find some other candidates that would retain the glucosinolates in leaves and reduce glucosinolates in the seeds. We do have one line that looks promising. We've sequenced that line, but we don't know what the mutation is. It's a very unique gene. So it's a very different, hopefully we'll find a gene in next year or so.
Okay. Matt Ott is asking, do you think there's a chance that stacking the three reduced dormancy genes could lead to pre-harvest germination under certain years or environmental conditions?
That's a
So,
good.
but in other words, before it gets harvested, are we going to have a pre-harvest sprouting or germination while on the plant, which is a problem in other crops?
Yeah, no, definitely it is. I could see it being a problem if you combine the all three, especially when you look at combining the reduced yellow seeded line, because you don't have that protection layer already. So you combine these other two genes and then you get rid of the seed coat, then it's very likely that you may have some pre-sprouting. But we have to combine these and test it before we can definitely say that's the case.
Yeah, there might be a delicate balance there. That's something that winter wheat breeders like Catherine Frells are going to they have to deal with because they're harvesting winter wheat in June or July, and then they're planting it again in September or October. So you don't want it to have too much dormancy, but enough dormancy that it's not going to sprout while it's still in the field, and you get these rainfall and misty days after it's harvest ripe. So it's a
Right.
delicate balance there.
Yeah.
Okay, Cody, I don't see any other questions in the chat or Q&A, and I don't think there's any hands raised.
No, I think that's it. Unless anyone else has any other questions, you can raise your hand or enter them in the Q&A chat box. Just give a second, maybe.
All right, well, I don't see anything else coming in, so Ratan, thank you very much.
We do. Sorry,
Very
Jim.
good
We do have one question here from John.
Hold on a second. Just to follow up to my question about seed size at deeper germination. It's interesting that you see an association between that with that in pennycress, because there was a study of like 6 or 7 legume species that used crop wild relatives, land races, and elite varieties, and it found that seed size wasn't closely associated with germination depth at all.
Right. So like I said, like this is just an observation. This is 1 year that we saw that it had larger seed. I cannot generalize that across, like I cannot generalize that having a large seed was always give us a good germination.
Yeah.
But the line that we see germinate better at a good depth is actually a bigger seed. But I agree that we're not going to see all the bigger seeds are going to germinate at higher depth.
Yeah.
Yep.
It's going to probably be variable across the species, how much
Right.
the seed size affects germination depth.
Right. Thank you.